Hello and welcome to this lesson for higher biology. This one is going to be focusing on key area 2 of unit 1 and we'll be looking at DNA replication. Prior to now we've looked at how DNA is organised and structured within the nucleus of the cell. What we'll be doing in this lesson is looking at how it is replicated, or we make copies of it within the cell. And in the next lesson we'll be looking at how we can also use a particular technique called PCR in order to make copies of DNA in a lab. So for today, what you're expected to be able to do is understand why and how DNA is able to make copies of itself. And you need to be able to state the need for DNA replication in the cell, list the 5 requirements for DNA replication, give the steps involved in DNA replication, state the role of primers, DNA polymerase, helicase and ligase in replication, and explain why we have leading and lagging strands forming during DNA replication. So to begin with, thinking about why cells need to copy their DNA, well, we know that DNA is copied during cell division or mitosis, and that really makes sure that the new cells produced have got the same number of chromosomes in them and they have the same genes on them. So really what we're doing is we're making sure that the new cells, the daughter cells, are genetically identical to our original parent cells, and that means that the parent cells can be replaced appropriately. Now, DNA replication is referred to as being semi-conservative. What that really means is the fact that each new DNA molecule is made up of half of a new strand and half from the original strand. So the original DNA is actually prized apart, new strands are formed, and these bind with the original DNA through complementary base pairing to form this semi-conserved molecule, giving one original strand and one new strand in each one. Now, when it comes to replicating DNA inside the cell, we need to have the original DNA template. We also need to have primers, which are short sections of DNA, and these are complementary to the DNA that is being copied. We need a supply of the DNA nucleotides with all 4 of the bases, so we need some adenine, thymine, guanine and cytosine available. We need specific enzymes, helicase, polymerase and ligase, and we also need some ATP, and that's going to give the cell the energy it requires in order to create the new DNA molecule. So looking at the steps of replication, we're going to begin with our DNA double helix unwinding. So if we were to imagine this as being our double helix here, we've got it running anti-parallel, and we would have our nucleotide bases coming in here with their hydrogen bonds. DNA helicase is going to come along and break the hydrogen bonds between the bases, and that really gives us 2 template strands for the other enzymes to get to work on. So in the next section, we have a primer coming to bind to the 3' end of our DNA template strand. This primer is complementary to this section, so it's just a short strand of DNA which happens to be complementary to this area. It'll naturally bind to it when DNA helicase has opened up our DNA double helix molecule. We then have exposed bases along our template strand. So here we've got our nucleotides, and you can see the bases are available, they've been opened out. That means that if there are any free nucleotides in the cell, they will appear and they're going to adhere and bond through their hydrogen bonds to those exposed nucleotides on the strand. So we've opened up our DNA, the primer has bound to the template, and then free nucleotides are going to come along and bind to the template DNA as well. However, that doesn't quite make our DNA strand yet. We need to create the bonds between the different nucleotides. And this is where our enzyme DNA polymerase comes in. It's going to come along and it's going to start attaching those nucleotides, the ones that have already bonded to the original DNA template strand. It's going to catalyse the reaction that forms a bond between those nucleotides, creating that strand. So essentially creating the bonds between the phosphate of one nucleotide and the deoxyribose sugar of the next. Now, DNA polymerase is particular in that it can only add nucleotides at the 3' end of the DNA. So if you imagine this little end section here would be the bottom part of that deoxyribose sugar, and the next nucleotide would have its phosphate ready and can only add the bond in that direction. And that leads us to producing DNA in specific ways. So as DNA polymerase moves along the strand, this is what creates our leading strand of replicated DNA. It's able to work in a continuous fashion, adding those bonds, creating those bonds between the new nucleotides as they've attached themselves to their hydrogen bonds to the template DNA. And it will only add them onto that 3' end, and that's what gives us what we refer to as being the leading strand of DNA. However, we also have an issue with this strand over here. It's not been replicated yet. And it's a little bit different. Because DNA polymerase can only add nucleotides onto the 3' end of the new DNA molecule, this strand has to be synthesized in small fragments. And as a result, this strand is known as the lagging strand, and it has a formation which is referred to as being discontinuous. Now, the long process of that, really very similar to the leading strand. We have our DNA, it's been opened up, DNA helicase has come along and broken those hydrogen bonds. The primer has come along and it's bonds to our complementary section. However, because DNA polymerase can add to this end, this is the 5' end of the primer, this is the 3' end, and DNA polymerase can only add on to this end, so the new DNA is catalysed here. Now that means we've got this section, but we need to go this way, we need to copy along here as well. So that's only going to happen quite slowly as DNA polymerase opens up the strand and lets more sections become available. What essentially happens is you have several primers coming along and binding, and DNA polymerase works down the way, adding new nucleotides onto that 3' end, new nucleotides onto the 3' end here, and new nucleotides onto the 3' end here. And that results in these shorter fragments of DNA being produced. Now they will then be joined together by another enzyme referred to as ligase, so that's going to stick those fragments together in order to form one continuous strand. And that takes ever so slightly longer because it has to be done in fragments, and as a result that's referred to as our lagging strand. So a quick overall review, if we're looking at DNA replication, you would see the original template DNA molecule being opened up. We would see the leading strand, template, and the newly synthesized strand being produced. So we would have had a primer starting here, attaching to these ones through hydrogen bonding as a result of complementary base pairing. Free nucleotides would have come in and attached themselves through hydrogen bonds along here. And then DNA polymerase would have catalysed the formation of the bonds between those nucleotides, making a new strand. With the lagging strand, as it's opened up in small sections, it would only have been able to create these little orange sections that you can see. The pink one is the primer. So DNA polymerase would only have been able to add the nucleotides along in this section and again in this section. And that results in several different fragments, which you might see, as in this diagram, are referred to as Okazaki fragments. You don't need to know the name of that one though. And just know that they're synthesized as fragments and then have to be joined together by the enzyme ligase. So from that, you should be able to explain that we need DNA replication in order to copy the genetic material of a cell so that new cells have exactly the same information as the parent cells do. That for DNA replication to occur, we need our original DNA template, we need primers and enzymes, we need ATP energy, and we need a supply of free nucleotides. That within DNA replication itself, we have the unwinding or unzipping of our DNA, the binding of a primer, and then the binding of free nucleotides to our template DNA, and then we have our formation of our DNA strand by DNA polymerase adding the new nucleotides to the 3' end of the new strand. On the lagging strand, we also require ligase, which is going to join the fragments together. The role of primers is really to give a starting point for DNA polymerase to work from. DNA polymerase's role is to add the new nucleotides onto the strand. Helicase is going to break open our original DNA template, and ligase is joining those fragments together. And the reason the leading and lagging strands form during DNA replication is because of that rule that DNA polymerase is only able to add those nucleotides onto the 3' end, and that means that the lagging strand has to be synthesized in fragments. Hope that's all made sense to you. As ever, get in touch if there are any questions.